Welcome. My name is Steve Hyman. I'm director of the Stanley Center for Psychiatric Research at the Broad Institute of MIT and Harvard. I'm honored to moderate this event for Keystone Symposia. As many of you know, Keystone Symposia convenes some of the leading life science research conferences worldwide. This webcast is part of their effort to bring more science to more people. As such, I would like to welcome the online participants who I understand come from more than 60 countries, as well as our studio audience. I would also like to thank Takeda Pharmaceutical Company for sponsoring this virtual event. If you want to participate in the discussion, please use the chat application below your screen or send us a tweet. What I'd like to do now is to introduce my colleagues on the panel. And first is Ben Neale, who is at the Stanley Center at the Broad and also at Massachusetts General Hospital. Ben.
Thank you, Steve. I'm a geneticist, and I work on really trying to identify what the genes and variants that predispose to psychiatric diseases really are. The genetics plays an important role in psychiatry, and it can teach us a lot about new biology and new findings.
Next is Guoping Feng, who is at the McGovern Institute at MIT and also the Stanley Center.
I'm a neurobiologist, and I study how neurons make connections in the brain and how they communicate with each other. I'm particularly interested in understanding how defect in neuron-neuron communication may contribute to psychiatric disorders.
And next is Stacie Weninger, who is a neurobiologist who is currently at F Prime.
Yeah, hi, I'm the executive director of the F Prime Biomedical Research Initiative. Our program supports a great deal of neuroscience research, everything from basic research in academia all the way through to translational neuroscience in the private sector.
Great. So that's a perfect segue when you talk about moving research into the private sector, obviously with the goal of new treatments. Now, I think many of our audience are aware that psychiatric disorders are common. The World Bank and the World Health Organization has reminded us that in aggregate, they're the most disabling disorders worldwide. And yet, there haven't been really fundamentally new treatments since the initial spate of discoveries in the 1950s that gave us antipsychotic drugs, antidepressant drugs, anxiolytic drugs, lithium maybe even earlier, and even some of our cognitive psychotherapies. So I'm wondering, what's different today? I mean, why are we actually having this panel in 2016?
So I think the first thing that's most different is that genetics as a toolkit is really starting to work. It's starting to work for schizophrenia. It's starting to work for autism. It's starting to work for major mental illness. And what do I mean by work? Well, it's really that we're finding genes and variants that we know are related to disease. Now, we still have a mountain of work to do to really understand what those biological consequences are and how we might go about interrogating the biology that these genetic findings are pointing us to. But really, we have darts in the dartboard, things that we can hold on to that we know point to biology that we know is relevant to disease. And that real causal arrow of genetics, that direction from this genetic variant plays a role in risk for disease unequivocally, is a really powerful and meaningful place to begin our interrogation of biology moving forward. And I think that's where what you do, Guoping, really kind of takes off from that sort of genetic finding level.
But Ben, let me not let you off the hook so quickly.
Worth a try.
So schizophrenia, autism, bipolar disorder, depression run in families.
Yep.
But we know genes are also not fate. So
how does it work?
How does it work?
So it's sort of, the way I like to think about it is actually how height works, okay? So if you think about your parents and you think about how tall you are, your parents and you are pretty similar in terms of your height. You're not identical, but there isn't like a gene that magically makes you 5'10" or 6'1". That's not the way that works. That's not the way height works. And the way schizophrenia works is much more like the way height works, which is there are environmental factors. You eat a little bit more or you eat a little bit less. That might make you a little bit taller or a little bit shorter. And there are genetic factors that you have a little bit more of this part of the biology, and that means that maybe your bones grow a little bit more or your bones grow a little bit less. And so these small perturbations, and they are small effects that we're identifying in genetics. It really is the case that these effects are small. But these small perturbations, they really point to what the biological processes are that are involved in the disease. And that really makes a difference if you're talking about something like Alzheimer's or even a psychiatric disorder like schizophrenia. These different parts of the biological system are available to genetics, and that's what we're beginning to gain insight into with the genetic analysis.
That's really helpful. So what we're learning is that genes are giving us clues to the molecular mechanisms that influence the risk of schizophrenia, bipolar disorder, other psychiatric disorders. Guoping, what do you do with that information?
Yeah, so that's a very good question. I think I'm really excited at this time is because of two major reasons. As a neurobiologist, the genetic findings is really make it possible to really go deep in cellular models or animal models to really dissect the biological reasons why these genes are involved in disorders. The other reason, exciting reason for neuroscience is the technology development. Now it is possible with the new genome editing technology, with the new expansion microscope, with optogenetics, you can actually probe not only the gene function, and you can put human mutations in cellular models and animal models to really see how these genetic mutations or genetic variations affect the biology.
But Guoping, I mean, I think I have insight into my pets. I think I know when my dog is depressed, but maybe I don't. It's hard to talk to my dog, and certainly a mouse doesn't get schizophrenia. So when you say you're using this genetic information with animal models to push it further, what exactly do you mean?
So that's a very good point. So we are very different from almost any animals, and evolution does a wonderful thing that add evolutionary pressures. So we diverge, develop functions and circuits that are appropriate, best for each species to survive. So whenever you use models, whatever the model you use, you have to take the evolution into consideration. That's the number one important thing. But at many levels, at the molecular level, cellular level, the gene function are highly conserved through different species. So at that level, it's animal model or cellular model extremely useful. One of the best example is from the genetic study of recently published in the C4, which is linked, is a signal in the GWAS study. But it's very puzzling because C4 is the gene involved in immune system. What's the relation with schizophrenia? Use animal models, study from Harvard and Stacie Center have shown this gene, use animal model, you can show not only the function of this gene in the nervous system, in neuron, but also show this gene is involved in pruning of neuronal connections, which has been long proposed as a potential mechanism in schizophrenia and as a development disorder.
Okay, so you're saying that these animal models, using genetic information, can teach us about mechanisms, but you're not giving mice schizophrenia.
No, right. I don't think we will never be able to diagnose any mouse as a schizophrenic or autism.
Okay, but that's important because a lot of people think that to be useful, an animal model has to perfectly model the disease. And so I think this is a very sophisticated place to be in. But how convincing is it? I mean, if your goal is partly to bring these basic research findings, whether it's from genetics or from gene-engineered animals or cells, where does it get us? I mean, most big pharmaceutical companies are still busily leaving neuroscience because they say it's too hard. So
I think that we are entering the stage where maybe it is no longer too hard, or soon won't be too hard. And I think some of that is because of the breakthroughs in genetics. The breakthroughs in genetics have given us real understanding of the disease pathology. They've also given us a real sense, in some cases, that if we hit that target, we will affect disease. And I think that's something that's been missing for a long time. Neuroscience research for a long time has been based on, in many cases, animal models that seem to have a phenotype, not based on human genetics, but seem to have a phenotype that we then ascribe to some neuropsychiatric disease. And we test a bunch of drugs and we see that, well, we're fixing this animal model, let's see if it works in the human. And we really don't know if hitting that target in a human is going to help. And I think that's why there have been a lot of failures too. So with genetics, we know if we hit that target, then we know we will affect disease. With an animal model, we can look at a lot of the molecular biology that's going on. We can understand the molecular underpinnings. It might even help us not understand the disease as a whole, but it might even help us with our quest for biomarkers, which I think is huge.
Okay, so we'll come back to biomarkers. But what you're saying then is, before we had these genetic clues, people would go to the lab and stress out a rodent and say, it looks depressed.
Yes.
What went wrong? But there was no anchor, nothing to tie that to the human.
Nothing to tie it to.
Right. And so now having genetics,
and sometimes it worked.
Yeah.
Sometimes it worked, and sometimes it. We've got the psychiatric drugs we have today, for the most part, from doing those kinds of studies. But I think we picked that low-hanging fruit, and now we really need to be more sophisticated about the approach, and we have the tools to do that.
So luck is not a plan, right?
No, luck is not a plan anymore.
So you mentioned biomarkers. So clearly, let's imagine, we have to talk about it more, but let's imagine that genetic discoveries and cellular and animal models and clever biology and biochemistry gets us to drug candidates. Mm-hmm. One of the reasons that the pharmaceutical industry is not so enthralled with psychiatric disorders is we have the DSM-5, right, where we say if you meet 5 of 9 criteria for 2 weeks, you have major depression, and that lifetime might affect 20% of humans, but we have no knowledge that underneath this big grab bag, even though this really serious illness, that people have the same things wrong with them. And then we have a clinical trial, and some people get better, and some people don't get better. Pretty much of a nightmare if you're trying to get a drug approved by the FDA and have it work in the population. So does the genetics help? I mean, how are we going to subdivide people?
Yes, so our current classification system does not reflect the etiological basis of these disorders by any stretch of the imagination. The kind of overwhelming results from the genetic studies show that a huge amount of the common variant risk factors, these common SNPs that are scattered across the genome that we all have, are shared across many forms of psychopathology. Schizophrenia, bipolar, major depression, ADHD, these phenotypes share genetic risk factors. And that means that our classification system isn't directly related to very precise biological perturbations. That these are much more kind of muddled phenotypes in a sense. And so one of the potential ways that we can maybe start to work ourselves to a more rational view about how we think about mental illness is to use genetics and the way genetic risk is operating in the population and use that as a handle to really move us towards phenotypes and kind of clinical classifications that reflect perhaps the genetic risk or perhaps reflect certain symptoms or certain biomarkers. It's difficult to predict what form that will take, but there's definitely this, kind of vista that you can see where these genetic findings can in turn teach us about what the real deficits are, and then we can use those deficits to really reclassify and redraw our lines for how we think about these kinds of traits.
Let me ask Stacie one thing, because I think if I'm right, you've supported a lot of Alzheimer's research, right? And so there was just this very pessimistic group of clinical trials failures where people were entered in trials based on the fact that they had dementia. Mm-hmm. And after the fact, it was discovered that a third of them didn't even have amyloid in their brains. They had Lewy body dementia. Right. But now there are, so what happened? What's different now?
I think the Alzheimer's field is a great example of where the progress has been made and things have changed. And I think there are a variety of things. First and foremost is we can now see amyloid in the brain of a human, a living human.
And we think amyloid's involved.
And we think amyloid is involved. So I believe that amyloid in the brain is involved in the disease. I'm not going to talk about which forms of amyloid or those kinds of things, but I think the genetics have pointed enough. APP, presenilin, you've got enough arrows all pointing to APP processing at least.
Better say what APP is.
Amyloid precursor protein. So a larger protein that gets cut to release this beta amyloid, which is what forms these sticky plaques in the brain, and it's what was a telltale pathological sign that up until about 12 years ago, I think could only be detected upon autopsy. So it was impossible to have a true diagnosis of Alzheimer's disease prior to autopsy.
Right, and we didn't want to enroll dead people in clinical
That's
trials.
usually not the best way to see if you can help someone.
Yeah.
So the ability to use PET imaging to see this amyloid in the brain of a living individual is a huge step forward. In these kinds of trials, the trials of today are not the same as the trials of even 5 years ago, where you would enroll someone in a trial based on their memory issue, having dementia, because we've learned that a quarter to a third of those individuals don't have amyloid in the brain. So if you have a drug that's trying to target amyloid, probably not going to work very well if someone doesn't have amyloid. So any trials going forward now, for the most part, are all enrolling based on positive amyloid in the brain.
So a biomarker.
A biomarker. And what this also does, which is, I think, so exciting, is that, in many ways, I think treating someone after they have Alzheimer's disease and trying to do something more than symptomatic is going to be very, very difficult. It's akin in many ways of giving someone a statin after the third heart attack. Yeah. Probably won't do that much good. So the question is, how do you identify those people before it's too late? We now know that amyloid starts building up in the brain at least 10, if not 15 to 20 years prior to symptoms starting. So there are now trials going on where they're actually enrolling people who don't have any real memory problems, but have amyloid in the brain to see if we can make a difference there. And we've got a biomarker that we're looking for. So I think that's a great example of the genetics pointing to something and then the technology coming along to allow us to be able to do more effective, more efficient clinical trials in a way. And then hopefully be able to see if you're having an effect. Now, the other problem in these neuroscience trials in general is how do you measure target engagement? And there have been
better say what that is.
Way too many trials where you're given a drug and you have no idea that the drug's actually even getting into the brain or interacting with whatever that drug is supposed to do. And it's really difficult in neuroscience. The brain is hard to see. It's hard to measure what's happening in the brain. So many phase 3 trials that have failed, after the fact, we realized that, we don't actually think that drug actually ever got to where it needed to get to do its thing. So we didn't even test the hypothesis that we were trying to test with that drug.
So actually, that's a really important point, right? Cancer is a really hard problem, but maybe it's an easier hard problem in the sense that somebody has an excisional biopsy and the surgeon hands the scientist is waiting there outside of the operating room, and you hand the person the disease. And in the human brain, there are very few opportunities to interact with tissue that is useful. And even still, we don't have diseases of single cells, right? The symptoms are exactly what the kind of thing Guoping studies, which are circuits. So maybe you want to say something about
I think
that.
This is a great point. So how do we develop biomarkers for psychiatric disorders? This is a really tough question, but currently, if you do a clinical trial in almost all psychiatric disorders, you have a questionnaire. You ask, let's say autism, you ask parents, how did your kids do in the past 2 weeks, right? They ask you today, it not only depends how the kids actually behave, it also depends how the parents feel. If it's a life stress and the boss was, asking
or
more
maybe
work
they're
to
just
do,
too hopeful,
and
right?
And the answer will be different, right? If you are, we're all parents. If you are stressed out, you are really tired, you think your kid's really annoying today. He just behave the same thing. The poor kid just because of you feel. So we really need, objective biomarkers. I think there are many different ways. I think it's a time, actually really good time to explore that. And there are many places, both academic and the industry, start to think about this. For example, you use autism as an example, but it's not. Many of the kids have sensory overload, right? Can we use this as a sensory response? For example, we call the ERPs, event-related potential. So you can measure from the outside the skull to see whether these kids are they combined with genetics, is there a subgroup of patients you can see an abnormal response? Not only this will be your biomarker to this a particular group of patients with potentially a particular subgroup of genetic mutations, but also can be used as a readout for efficacy of treatment.
So this is a really important point, and I wonder, Stacie, as you're investing in this kind of research, how do you weigh diagnostic biomarkers, you're in this bin versus that bin, versus markers that change with treatment.
Right, they're agnostic, as we would call them. So
Yes.
I think that they're both critically important. So we weigh them both equally as very, very critically important. We want to make sure that we're enrolling the right patients in a trial, and then we want to be able to measure whether the drug is actually having an effect as we would hope it would.
So maybe a really good example of that is viral load in HIV, right?
Absolutely.
If the drugs are working, the viral load plummets.
Right. You need to be able to measure that. And I think we need to be able to do effective and efficient clinical trials. And that is a problem in neuroscience in general, that we do these very, very, very large trials where we ask the parents, how's the kid doing today? Or they're based on these fuzzier outcomes,
Okay,
which
so
gets a lot of
it's worse
variability.
than that, right? You're saying first we do these trials based on the kid's symptoms, but
Right.
the kids could have lots of different causes of
Absolutely.
those symptoms. So it's a very heterogeneous group of people.
Absolutely.
And then we have to ask the parents, or for schizophrenia, we have to interview the person
Yes.
from time to time. So that's maybe not so attractive if
It's
you're
huge.
a company.
And
Yeah.
I think that's where the genetics play a huge role here, in that with genetics, in some cases you can subdivide, and in some cases that subdividing even gives you a sense of what therapeutic might work. And so you don't want to test your therapeutic in the general schizophrenia population or the general autism population. You've got a therapeutic against a particular target that you think is disrupted in a particular set, and then maybe you develop this biomarker that would broaden it to the general population even, of, okay, well, if we look at the general autism, if they have an ERP abnormality or something like that.
So kind as she is, I think Stacie's just put you on the spot.
I agree, but I think genetics can go one better. So I think it's not simply this question.
He's taken up the challenge.
Not simply this question of can we subdivide patients and can we stratify them based on their genetic risk landscape and do that kind of analysis. I think genetics also serves the purpose of understanding whether those intermediate phenotypes or those endophenotypes that we hypothesize about.
What's an endophenotype?
It's so an endophenotype is kind of got a long and storied history in psych, actually, I think is where it really originated. And it's this idea that there's something that you can measure that stands between your kind of basic genetic landscape and then the outcome disease. And that intermediate phenotype plays a role in risk for the disease, and as a consequence of being kind of a causal intermediate phenotype, if you target that intermediate phenotype, you can actually have an impact on reducing the risk in disease. Now, the most natural example of this is lipids and myocardial infarction or heart attack. So the reason that we give people statins is to reduce their LDL. Reducing the LDL shows a reduction in myocardial infarction. Now, that's been shown not only epidemiologically, a la Framingham and many other kinds of considerations, but it's also been shown through genetics. So if you look at the genetic risk factors for LDL, and then you look at those same genetic risk factors and ask whether or not they influence heart attack, they do. And that's not the case for HDL. If you look at the genetic risk factors that are specific to HDL, they don't bleed through and show this kind of risk associated with heart attack. And the reason that's important is that both LDL and HDL correlate with heart attack in the population. So if you measure someone's HDL, that is a risk factor. It's the same thing with C-reactive protein. So CRP is exactly the same story as HDL. These are great clinical markers. They're really useful in terms of saying, are you at risk for something? But they're not useful for therapeutic intervention. And that's where genetics is really critical, showing that there is the same genetic risk factors on the intermediate phenotype that you're looking at, as well as the outcome phenotype, gives you a much stronger sense that this is a worthy target for therapeutic intervention.
So it's all about following the causes.
Yep.
And not simply falling for the correlations.
Exactly.
It turns out I can raise my HDL by exercising and then drinking a few glasses of red wine. And that might influence my risk of heart attack in a positive direction, but it doesn't mean you can manipulate my HDL.
Correct.
Right?
Exactly.
It's just a proxy.
And that's why genetics is really critical. Not only is it teaching us biology, it's in some ways acting as an arbiter for whether or not something's relevant. And that relevance is, I think, lack of efficacy is the major reason that we're not having success in therapeutics right now. And that's why we're so enthusiastic about the progress we're making in genetics, because it helps to table the whole conversation.
That's good. So not that I was suggesting that the way to quench your thirst after exercise is with red wine, but Guoping, can you, do animal models help us get to endophenotypes, biomarkers relevant to humans?
Yes.
Sometime?
Yeah. So I think in sometimes, yes, I think animal model will play a very important role. The reason is our brain is very different from almost any other organs. So they do not, the cells in the brain do not function autonomously. So you cannot just simply take a cell, a blood cell, red blood cell, you transport, oxygen, but the brain cells, they have to communicate with one another, and so form a particular circuits. And these circuits output actually is control your behavior. So we are try to treating correct behavior abnormalities. So understand, I think in the neuroscience in the brain, these endophenotypes can be measured by neurophysiological markers. For example, even in the cells, right? In cultured iPS cell-derived neurons, or in animal models, to look at how this mutation affect the neuronal function. And if you correct the neuronal function, that function is not a readout of the behavior, but it is the underlying mechanism of that behavior. If you correct that, and that will give you prediction that it will be therapeutic, right? So in that sense, actually, these are real biomarkers, endophenotypes that actually can help to understand the genetic landscape, because genetics is very heterogeneous for almost all the psychiatric disorders. But use these cellular neurophysiological phenotypes, you can actually subgroup the genetic effect. Actually say this group, although the genes look all different, they all affect this type of cellular neuronal physiology. That's the output, and the treatment selection should be very similar.
Okay, so you want to use genetic information, find some physiologic difference related to those genes, perhaps in an animal model, and see if you can push that, tweak that in a positive direction, and that would give you more confidence to go ahead in humans.
Exactly, yes.
So there are a few ways we could go here. One is to say, how much can we really rely on animals, and how much do we have to make progress in human experimental biology? But maybe even before we get there, I would say, this is a great conversation. There's hope. Genes are being discovered for the first time. But where are we? I mean, in history, what do we know that's really actionable now or in 5 years or 10 years? Because I'm sure there are people listening. Look, every family has these terrible illnesses. And so where are we?
So on the genetics, we're still at the beginning. And I know that's kind of a funny thing to say. There's been a huge amount of excitement about what's happening in genetics.
How about the end of the beginning?
Yeah, maybe the end of the beginning.
Yeah.
I think that was that was what was kind of on the cover of Nature for the first, kind of the final report of the 1,000 Genomes Project, this kind of effort to kind of start to collect common and rare variation in global populations so that we can understand what it looks like and how sequencing will work. And the sample sizes that we put together now for genetic studies are unprecedented, almost in any kind of walk of kind of investigation. 37,000 cases of schizophrenia recently published. That's up around 50,000 now that is in the kind of pipeline and being analyzed. And that's a huge sample size. That really is a tremendous sample size. But we still have a tremendous distance to go. We found 100 loci. There are probably another 500 or even 1,000 that remain to be found that are also important players in what's going on. And that's just schizophrenia. It's not that all of mental illness can be compressed into schizophrenia. It doesn't work that way. There are lots of different kinds of mental illness that we need to focus on.
Wait. So if we have 500 or 1,000 genes with little tweaks in them contributing to risk, how does that work?
How does the brain work, I guess? Maybe it's a question for Guoping, right?
But but seriously, how do we really profit from that knowledge? How does it come together?
So the first question that's always asked after you found a new genetic hit or variant is kind of what direction. So if it goes to a gene, is it upregulation or downregulation of a gene? And that matters. And then the other questions you start to ask are, what does that gene talk to? What does it interact with? How does it exist in the biological system? And what are the kind of mechanisms? behavior, cognition, these are very complicated phenotypes. It's not like, walking, lots of animals walk. A whole bunch of things have movement, but not everything has, consciousness, right? So these are the kinds of things that we're talking about at a biological level, and I'm not surprised it's a little more complex in that landscape than it would be otherwise.
So one of the things you said is what gene talks to what. So is there some convergence on a smaller number of pathways? Do we know that? Guoping, what do you think?
So I think if genetics is at the end of the beginning, we are at the very beginning of the beginning.
Well, that's
so,
optimistic. Yeah.
But
Is that
I would
a message to federal funders that they got to keep going?
But I want to give the signal is we are at a very exciting phase of the beginning of the beginning. It's really exciting because.
Well, you remember the beginning, right?
Yes, you will remember this beginning like your first kiss. It's a very exciting beginning.
A true scientist right there.
The reason is that these genetics, although they are very heterogeneous, the new technologies allow us to interrogate thousands of them at the same time and looking for commonality. What do they converge on? I don't think they will converge on single mechanisms, but just by the data analysis from genetics already pointed a few major pathways. One is the synapse, right? Synapse is the small structure neuron communicated between each other. So it totally makes sense because
So wait. So you're saying lots of genes that we're discovering that are related to this illness, schizophrenia or autism, do something particular in synapses?
Yes, they already show, these genes are already identified by basic neuroscientists that show they are important for neurons to make connections, make communications. And neuron communication is the building block of our brain communication.
Sure, how our brain computes.
How they compute. So it completely makes sense, right? Now it's our job to figure out how these mutations affect the neuron communication, how does the neuron communicate, in what cell types, in what particular circuit in the brain. Once that's identified, it's not far away that we can identify specific targets for this group of neurons that we can modulate, because the new technology like DropSeq, which is you can sequence you can profile single cell, tens of thousands of single cells RNA expression that allow us very quickly to identify what are the unique druggable targets in each individual cell types. So once neurobiologists figure out what is the exact defect, the direction of this defect, we can actually move on to.
Okay, but before I get on to the human model for the human, and I just want to come back to Ben. So you're understandably very excited about what you can do, but Ben is, I guess I offered it only at the end of the beginning, and saying that we need a lot more patients to find these genes. So give us a scope out. What is the project? If we want to understand the genetics of schizophrenia, bipolar disorder, autism, what are we talking about in terms of the number of human subjects to be genotyped? How long do you think it's going to take? I mean, you're already feeding the information to neurobiologists, but give us a sense.
So I actually think of it not as a kind of one and done scenario. I think of it more, to kind of harken back to what I was saying earlier, as this feedback loop where we start to identify these genetic risk factors, and then that starts to teach us about what parts of the biology are relevant, but it also gives us some insight into maybe what parts of the human phenotype is being influenced. And then we take those and we go back and we look at the humans and ask, how are you doing on this kind of phenotype? How do we reconstruct our measures? How do we rethink our approach to the way we're classifying and treating patients?
This is good because it means that if you sort of have a pretty complete story of the genetics of schizophrenia, you're not out of a job, and so you actually have an incentive to keep finding those genes.
Well, I think it's just because what we call schizophrenia is not a single clinical entity.
Right.
It really
the
isn't.
DSM is really
and the biology doesn't respect the DSM. It seems to think it's not classifying things appropriately.
Right.
The other thing that I would say that we need to make sure to hit is lots and lots of different populations. So the kind of genetic risk factors that are active in European populations are not the same necessarily as those that are active in Asian populations. Certainly in terms of frequency between those populations, there's a lot of genetic risk that is shared. That's not going to go anywhere anytime soon. But there's a lot that's specific, and there's a lot we can learn about genetics from taking a much more global perspective. And so while we have 30, 40, maybe 50,000 cases of schizophrenia from European ancestry, I'd like to see us do exactly the same thing in a cosmopolitan way, because I think that will teach us not only about what's relevant in those different populations, but also mean that when we start to get to therapeutic hypotheses, that it's not strictly for the set of individuals that have gone through these kinds of studies already. And I think there's a moral component to this.
Right, there's a global equity issue.
Absolutely.
So that's good, both science and global equity. Okay, so, but you're optimistic, right, Ben? We're going to succeed?
Yes, I think that, the fact that it's working now sort of means that people are like, well, it's worked and it's done, and that's not really true, but the fact that it's working is true. And I think we're always eager and enthusiastic to turn our attention to the next new exciting technology or approach or consideration. But I think there's an awful lot of work still to be done in scaling up the genetics effort to really get us to a point where we have a much more comprehensive picture of at least the genetic risk factors that influence schizophrenia now. If you tell me there are two kinds of schizophrenia, and you've kind of jumbled them up together, we don't have enough information to delineate between those two kinds at this point in time, I would say. And that's an important payoff.
Good, good. So you're going to keep at it with your colleagues globally, and Guoping is, putting it to work in animals. But I think I've heard you say, maybe I sort of said it also, that we're not going to have an animal with schizophrenia. So ultimately, if we're going to, if therapies, if medicines come out of this research, we shouldn't decide whether it works or doesn't work only in a mouse.
Yeah.
So what are we going to do?
Well, absolutely. I mean, I think the excitement of what is yet to come in genetics, there's so much more promise behind, but we have enough to get started. Right. And so I think as we're getting started, and there are animal models now, and the way we're using the genetics to make relevant animal models, not a mouse with schizophrenia, but to look at these synaptic defects and to look at exactly what's gone wrong, and then to create some hypotheses about how to fix that. So, earlier I was saying genetics might give you a target that if you hit it, it's going to affect humans. I don't think that's quite where we are in psychiatric research. I think we have targets that allow us to interrogate circuit disruption, which allow us to form hypotheses about what kind of a target might work. We use the animal models to test some of these hypotheses and to see if our target that we have chosen is going to affect that circuit disruption, that synaptic disruption. But ultimately, the test has to be in humans to see if we can affect disease.
Let me get this. So, first of all, you didn't say it, but of course I know you agree that we have to show that any chemical we're going to put in a human is safe,
of
right?
Course.
And we're going to do all of that. But assuming something is safe, we're going to go into humans with an illness to see if it works, even though we just can't get that answer in an animal.
No, we have a sense, we have a strong hypothesis that we think it will work based on the fact that our animal model was grounded in human genetics, and we identified a disruption in the animal model at a circuit or a synapse, and we've corrected that with a molecule that is safe.
Yeah.
And it's a molecule that we know is going to get into the brain, and we know it's safe at the level to get into the brain to affect its target. So that's very, very important to say that this is, we're not going, and that's what these early studies are about. We're not guessing. We know if we have a compound that is going to interact with target in the human brain at a certain amount, that we will affect the circuit based on what we've seen in the animal model. We think will affect disease. That's how to design the trial. Now, the question is, what's the readout? I think, as excited as I am about the field, I think the idea that we're going to have a molecule that fixes an autistic kid or fixes schizophrenia in totality is difficult. But I think that it is within reach.
Not once the illness is established.
Maybe earlier. Yeah. But I think where we are right now that is exciting is that I think we have enough idea of what's going wrong in some of these diseases, that we will be able to significantly affect certain aspects of the disease, which will significantly improve the quality of life for these patients and their families. And so I think understanding to fix what aspect you think your molecule is going to fix and designing your trial to measure that, not necessarily by, gee, how was your kid doing for the last week, but really measure in a concrete way.
Let me try to make this a little bit harder for you. I actually have had a lot of conversations in various workshops with FDA, and they buy this. They just care that the medicine is safe.
Right.
And they remind us that in cancer, a lot of drugs really only can go into patients because of toxicity.
Right.
It would be new in psychiatric disorders. But how about a company making a financial decision? I mean, what you're saying is, we're pretty sure, based on the genetics, based on the kind of work Guoping does, that we can fix this defect in a synapse. The company's going to probably want to know, but is it going to make a difference for a patient's symptoms, right?
Well,
and you're not going to know that necessarily. So do you, I mean, in some ways, you invest in people and projects. Do you think companies will have the courage, the financial courage to invest on that basis?
I think that having the animal models grounded in the human genetics is what is giving people the comfort level that they will affect the human disease in a way that has not been done before. This is not just, does a random compound help a mouse in the forced swim test or something like that. This is really based in, we're not trying to model the whole disease, but it's based in the human genetics that gives us a very strong hypothesis. We don't know till we do the human study, and that's absolutely true, and that's why
always true.
It's always true. And I think that's why we need to figure out, because these studies do cost a lot of money. It's very expensive, and we don't want to expose a lot of patients to a drug that even we believe it's very safe. No drug is inert. If it's inert, why develop it? So we want to be sure that we're being very rigorous in how we're choosing who to expose and what we're measuring, and make sure it's working before we expand. So I think some of the smaller, almost proof of concept sorts of studies. I mean, I think the intramural program at the NIMH is a great example of really trying to develop rigorous biomarkers and doing much smaller studies that would not be the basis for approval, but maybe the basis for understanding, do you go forward with a larger study? And I think those are very powerful and really helpful in this field.
That's good. Did you have a comment?
These intermediate phenotypes are, in some ways, the white whale of psychiatry. I mean, we've had this idea for 50, 60 years, and we're not there yet. I think, it's not to say...
We want a better outcome than Captain Ahab, too.
We do. And I think that's kind of implicit here. But I'm not so negative that we won't get smarter about the way in which we try and do these things.
Good. One thing which often we don't like to discuss, but as the pharmaceutical industry has moved away from psychiatric disorders, and really most neurologic disorders, most brain disorders. I mean, epilepsy is also terribly underserved. The question, and psychiatry has not had this kind of handle on mechanisms. We often haven't had the right people engaged in this translational quest for real therapies. Is it too early, or are you Guoping, are you seeing people entering this field, young people who kind of get the modern biology and the genetics, who are interested in therapeutics? Because I think that's got to be part of the revolution.
It's a critical. From basic science field, we have graduate student, postdoc. It's very, very attractive to them because they see now it was a black box. Now you actually can see that it's, to me, like with the technology, with genetic. I told all my students, this is like a scientific playground. You can really, you have a question. Now you have a set of tools. You can use this tool to solve the problem. What's better than that? So it's a great field. On the other hand,
because even Dr. Alzheimer once called schizophrenia the graveyard of neuropathologists. So what you're saying is
not anymore.
Not anymore.
Not anymore.
Yeah.
I think it's literally it's like a heaven made for a playground, basically for scientists to really figure out the problem.
But before you, I want to ask Stacie of the more. I don't know if you're seeing this the sort of a stage later, in development. Are you seeing sort of a change yet in the kinds of people, or is it still kind of early?
I mean, I think that there's definitely building enthusiasm, and I think that there are a lot of scientists even who maybe were in other areas of development who are starting to see, well, neuroscience is pretty exciting. There's a lot going on here, and applying that.
Mostly for biotech and more adventurous companies, not the big.
I mean, I think that's certainly what I see more, but I think that I think there's a lot of excitement, and I think people are gravitating towards that excitement.
Because without the people, the science.
But one area we really need more young people is training new psychiatrists and link them to the new era of discovery and neuroscience. So we can work together to move the basic neuroscience to the clinic. And for any clinical trials on any of this type of human study, we need the new generation of psychiatrists to really understand, based on the genetics and neurobiology, not only based on DSM-5. So how to build this, there are many places start to build this kind of program. That's a really critical thing.
I mean, I have very complex feelings about the DSM. I mean, on the one hand, they give us some structure, and while there's a lot of genetic risk factors that are shared across, say, schizophrenia and bipolar, or between bipolar and major depression, there's specificity as well. There are things that are very clearly not shared across these different classifications, at least as it stands now. And so it's sort of gotten us started in this kind of quest for understing the genetics and the quest for understanding the biology.
But Guoping wants to kiss it goodbye already.
Yeah, exactly. So I mean, and there's all of the kind of clinical activities that it serves as well. It gives some sort of common language and those sorts of things. So it's not as if it's utterly hopeless, but it's also clearly not suited to what it's targeted at. You can contrast that with how we think about the classification of neurological disease, and how we want to put the premium on amyloid for Alzheimer's. I mean, that's a fundamental difference in what we're measuring and what we're looking at to define these various illnesses. And I think that's, that contrast is partly because of the challenge in dealing with mental illness in general.
So in fairness to the people who put together the DSM, we just didn't know enough, and we had to make these guesses based on symptoms. Maybe the problem is that instead of sticking to the 10 illnesses or so where there was a lot of research and a lot of knowledge, they had to support the entire clinical enterprise. You ended up with a book with almost 300 diagnoses, and some of them surely don't map onto nature.
That's a safe bet.
Yeah. So.
But we don't know enough of neurobiology of this disorder. So in the future, this will gradually, slowly improve and change and
that's exactly the feedback cycle.
So what you're saying is, DSM-5 schizophrenia was good enough to get us genetic results, and maybe it's inexact and noisy enough that it actually increases the number of people that you have to enroll, in order to get a signal. But the key is whether people will be listening to the feedback and iteratively improving the diagnostic criteria, discovering and validating biomarkers of different flavors of illness so that people can be treated well.
Yeah, I mean, these are not
hard
the
things.
Spoken word. We shouldn't treat it like the spoken word. And that's, I think, sometimes the temptation in clinical environments is to do so, and you can understand why they make those decisions, but I don't think that reflects the true root causes or the biological processes that we're tapping into.
Good. How about the biotech sector? So we've talked about big pharma walking away. Certainly a lot of people in biotech and people in venture who have their ear to the ground and their eyeballs in, some of the local labs, see what's happening. Any more interest there?
Oh, absolutely. And also, just to be fair, not all big pharma is walking away. There's definitely still a lot of great research going on in big pharma.
But it's just the aggregate movement.
Exactly. But I think that in in biotech, in neurodegenerative diseases, I mean, we've been involved in a biotech that's recently launched that, that is an example, I think, of the resurgence of interest of investors in coming back in to the field. And in believing that because of the biological advances, the technological advances, that we really are going to be able to make a difference. I think in psychiatry, it's maybe still a step behind that, but it's certainly coming. I mean, again, we've been involved in some biotechs that are doing some psychiatric research as well, and I feel like the area is heating up, that there are more and more people that are starting to think about how can we translate these things and how can we get therapeutics, and have some ideas that they're trying to test now. So I think it's heating up. I wouldn't say it's hot yet, but we turn the fire on.
Okay, so we've been talking, really because of the genetics or molecular clues, we've been talking about drugs mostly. But one question I wanted to ask, and I know some questions have already started to come in, and I see somebody's even asked about it as well. These are genetically influenced diseases. What about genome editing with things like CRISPR-Cas9 or son or grandson of CRISPR, which has already been published? What about, delivering gene therapy? Is that relevant, or is that more relevant to other kinds of illness?
It's a mix. There are certain mutations that predispose very strongly to intellectual disability, to forms of autism. There are these so-called Mendelian diseases that have, effectively a single gene major driver. It may not always give you the disease, but it will certainly give you a very strong probability of having such disease, like right. 4 in 5, something like that, or even 1 in 5, and it's pretty bad news. And the kind of, I'm not an ethicist, so the question of whether or not we should engage in gene editing for that sort of thing is probably more of an ethical question, conditional on safety and efficacy and the time at which you need to intervene, given the therapeutic discussion we had earlier about whether it's developmental or it can be palliative at the end. But those are the kinds of mutations that I think there will be like the most obvious path forward with respect to that potential therapeutic set of avenues and considerations. Most of the other mutations that are identified, in most instances, these common variants, we don't know what the variant is that's relevant, and the effect that it's having on the disease or disorder is very weak. It's very subtle. It's maybe increasing your risk 5 or 10%. So moving you from like 1% to 1.05% chance of having schizophrenia. And so I think that gene editing for those kinds of mutations is off the table, at least at this point in time, until we understand much more clearly what the consequences of these genetic variants really are, and then we can start to consider it as part of the range of potential approaches.
Okay, so what you're saying is that for common-ish, 1%, common enough, common polygenic diseases like schizophrenia, genes give you really important clues to disease mechanism and potentially biomarkers and therapy, but by themselves are not sensible targets for gene therapy.
Whereas like Mendelian forms of intellectual disability, things like ARID1B, which is a gene, knock it out, it causes intellectual disability, that may be a much more sensible line of interrogation.
Right, and people are really focused already on some adult neurologic disorders caused by single genes. So that may be in the future.
I think like, some of the neurodegenerative disorders, whether it be Alzheimer's disease, familial Alzheimer's disease, where there are these genes that are single mutations with 100% penetrant, that may be, again, I don't think this is the panel to discuss the ethics of whether or not that should be done, but I think that if it were to be, these would be the kinds of things.
But like Huntington's disease is a very good example, right? You can do genetic tests, when the expansion, CAG expansion, how long, and you can predict in how many years this person is going to have this disease, and within 10 years onset, this patient is going to die. You can predict. So these are the perfect examples. Single gene, know exactly what the mutation, what exactly you want to do. You just want to shrink the expansion, and these cells will be fine, and the patient will be fine. So this might be the best example to go.
I think where it gets, you start down your slope of very low penetrant, something like ApoE. ApoE has a huge risk in Alzheimer's disease, but it's not 100%.
Yep.
So that I think is where the conversation, when the single mutation, 100% penetrate, to me that's an easier one to think about. And then you've got some that are high risk, but not 100%. And then you've got a whole myriad that I think I wouldn't touch because the impact on disease is just too small to really know.
And in some ways it's a race, right? I mean, in the event that we develop therapies or targeted small molecules or those sorts of things that can address the biological changes, then we don't actually need to engage in the genome editing,
that would be the
upfront,
preferred route.
right? Like in a sense. So that
it's a great point.
Yeah.
So I'm going to take questions in just a few minutes. I'm just wondering whether there's anything any of you really wanted to say that you didn't get to say in this panel.
I want to mention one thing. We talk about animal models. The models does not, is not limited to animals. And we have the new technology, induce pluripotent cells, which you can take a patient skin cell and reverse it into stem cell, and then you can reinduce them into neurons. And that's actually a really important tool for study polygenic disease, because we don't right now for schizophrenia, most of polygenic small risk factors, we don't know exactly what the combination make this patient successful to become clinically schizophrenic. But these cells, actually, these cells have the perfect bad combination to be pathogenic. So you take the cell from the skin cell from the patient, you can induce it into neurons, and you can study what are the endophenotype, what are the cellular defects in these cells. That can be developed as a cellular system. Not only can identify converging mechanisms, but also this cell can be used for screen compound. What drugs you can use to correct these biological problems.
I'm going to turn to the studio audience in just a minute, but somebody's tweeted a question which I can't resist right now because of what you've been saying. This person asks not only about human cells, but also human organoids, the ability to grow, to coax any stem cell you like with any genotype you like into a little liver, a little kidney bit, or a little bit of forebrain.
So this is a very powerful tool. The organoids is a culture of these stem cells. You can grow a miniature of structure that contain some features. For example, cortical layers, and they have different type of neurons. Our cortex are and some of them can be actually developed in vitro, and this will be very good.
Are they going to replace animal models?
No, they're not going to be. But they actually can replace some of the animals for study microcircuits within a small regions, right? Because these organoids don't have blood vessels. They don't grow. We cannot grow a brain.
Better. But I guess what you're saying is cells, organoids, animals, humans.
Working
depends on what your question is.
Exactly.
Right.
Yeah.
We need them all.
Right.
I think that's what's important.
Okay. All right. Anybody with a question, please stand up so first of all we can hear you, and someone will bring around a microphone. I'm also getting questions sent in by Twitter, and anyone. Let's turn to our studio audience first.
I actually have two questions. The first is related to what you said at the beginning in the discussion about the fact that it's actually small perturbations. So given that, if you identify the 500 loci, let's say that it goes down to 10 pathways, what's the right therapy? That's the first question. Do you want me to ask the second, or do you want to finish this first?
Why don't you ask both? And
So the second one actually relates to you, which is, let's say you do have the profile and you know exactly what an individual looks like and what those perturbations are. Do you think that industry is comfortable with stratifying patients and saying, okay, we know that this number of people have this profile and that's the only ones we're going to treat? Because having worked at FDA, the applications tend to be broad.
Mm-hmm. Okay, so these are great
Excellent.
questions. Yeah. So do you want to?
Sure.
Yeah.
So on the, maybe there are 10 pathways, which one do you pick, or how do you pick a winner? We don't know. In some ways, the hope would be that these pathways would point us to other things that we could measure so that we could understand what your risk landscape looks like. And again, just to return to heart attack, because it's a convenient analogy, if you have really bad lipids, we're going to target your lipids. If your lipids are excellent, but you have a family history of heart attack, we might maybe monitor your cardiac rhythm a little bit more aggressively and take a look at that. And those are not the same pathways by any stretch of the imagination, but both of them are clearly relevant to heart attack and kind of those kinds of disease endpoints. And I guess my philosophical approach here is that in, say, for schizophrenia, if there are these 10 different pathways, maybe some of them will be able to pick off by intermediate phenotypes that we can point to and say, this is where we want to intervene therapeutically in the patient. Or we'll do what we do now, which is we design against, we try our psychopharm against patients and see what works. And it's not obviously the way we'd like to do it.
Target multiple pathways at once. Although
exactly.
For FDA, that's a bit of a nightmare, right?
Naturally.
Yeah.
No, and I think to your second question, it's an excellent question, and I think that the field has evolved tremendously in this over even the last 10 years, if not a little bit longer than that. And that, it used to be that the pharmaceutical industry really didn't want to touch orphan diseases because the numbers were too small to justify the investment that would be required to make a drug. And I think that has really changed. Many companies are now all about just orphan diseases because the clinical path forward might be easier and more efficacious. And so I think I don't want to bring up the political topic here, but I think that with appropriate pricing, you can get a return on investment, even given very small patient sizes. So I think that if you could convincingly determine that if you stratified these patients based on this endophenotype, this genetic, this something, that you think that your therapeutic would be affected in this subset, it might be much, much, much smaller than the general schizophrenia or the general autism. I think that there is a path forward if your drug has a significant effect. And I think that's also key, that you will never command the price to get your return on investment if you're barely affecting the phenotype. But if you really have a game changer that really changes the quality of life for someone, then I think you can get the return on investment to justify that. And I think that companies have realized that and are willing to put the investment in to even small numbers of patients.
Here we have two. Go ahead. Get a microphone.
Thank you very much. As you think about your genetic and biological clues and looking ahead, are we thinking about psychiatric disease as sort of a global brain disease, or are we really talking about fixing specific regions or circuits or networks? And if so, do we need to be thinking differently about how we might apply the therapies of the future so that to specific brain regions or areas?
So I took away that we're trying to stratify people genetically and we're going to target therapies to particular synapses and circuits, right? And that people with the same diagnosis may actually have different underlying things wrong. I mean, this is the hope of precision medicine, which right now is much more close by for cancer, where you sequence a particular somatic mutation in a tumor and match someone with the right therapy. There are other problems because tumors are heterogeneous and you give a drug, you're playing Darwin and other things escape, right? I mean, but here we're not.
For the most of common psychiatric disorders, this will be the case. Also, there are maybe some other very severe early neurodevelopment disorders that the gene expression is very early. They are critical for a lot of things at the very beginning of development. Then that will be the case, but that will be not for most. Think about schizophrenia. Most kids grow up, functions well, go to school, even go to college. So this will be the case, but there will be rare, much smaller cases that affect the whole brain. This is more severe intellectual disability kind of thing that's much more often.
But for these other conditions, I think you want to focus on particular synapses and circuits.
Yes, exactly.
Thanks.
I think it's been abundantly clear that the new exciting data generating excitement and driving the new investment into neuropsychiatric drug discovery comes from the human genetics. So my question is, what resources can be brought to bear to further dig into the genetics to better define not just the best targets, but which gene is the target, which direction the target should drive, and how common those are, and how they correlate with the human patient population, because these are human phenomena. So I think we can launch those ships of drug discovery when we feel confident enough in an individual target. But what else can we bring to bear from a community perspective on the genetic definition of those targets?
It's an excellent question. Our ability to understand what the genome is actually doing is still pretty poor. I think when the Human Genome Project was completed, Eric Lander was asked to kind of sum it up, and he said something to the effect of, the Human Genome Project bought the book, hard to read. And then I think that kind of captures the challenge that we have right now is, we've now identified genetic variants. We're confident that these variants are relevant to the diseases and disorders. And the struggle and the challenge right now is what other molecular biological techniques can we start to use to interpret how the genome is behaving biologically? How is this genetic variation influencing the various components of things like epigenetic state? What DNA is exposed and what isn't exposed? What's getting kind of translated and what's not getting translated, and transcribed and not transcribed, and all of those kinds of biological operations that the genome is engaged in are starting to come into focus. So things like the Epigenomics Roadmap project and those kinds of things are teaching us about kind of a coarse resolution of what is relevant, but we're a long way away from being able to say, well, these variants, they're going to change your epigenetic regulatory state in this fashion, and that's going to knock down the expression of this particular gene in this cell type by this amount. And that's not, we're not there yet at all with respect to that kind of landscape. And maybe we will get there, but that's like one avenue that you can see in front of us that would improve things.
So, Ben, the complexity of this points
really
to
bad.
no, yeah, it did get really but there's a great question. So while the studio audience is sort of dreaming up other great questions, there's a great question somebody sent in. How can parents, and I would add not just parents, but family doctors, practitioners, other mental health practitioners, how can parents get up-to-date, reliable information that can be understood by non-scientists? And Stacie, you were involved, at least in some way, with Al's Forum, but
Yes.
is there, the trouble, there's a lot of, you go on the web and
There's so much information.
what's good information?
Al's Forum is a great example. It's something that I run, which is started 20 years ago, and but it is for Alzheimer's disease researchers, and that is the target audience. Now, it provides news. Anytime a new study is published, there's a conference. We have a team of science writers that write a story about it. We certainly have many, many members of the general population that come, educated lay, who will read the stories on there, and this is specific for Al's Forum. Now, there was a sister site that actually one of the managing editors of Al's Forum left to start, called the Schizophrenia Research Forum. Again, it was really targeted towards researchers, but certainly educated lay come there, and there have been several other spin-offs of that. And I think, trying to have more involvement of scientists in actually sharing the latest state of the art and the knowledge with the general public, I think is important, because I think there is a lot of information out there and there's a lot of misinformation out there. And so trying to create some reliable sources, and I think it is up to the entire scientific community to play more of a role in communicating their scientific findings to the public in ways that's useful.
I think this is a critical, important. We'll not name names, but if some of the diagnostic proposal is just not ready for genetics.
Yeah. Okay. Do you have a microphone? Okay.
Good afternoon. My name is Stephen Alsop, and I'm an MD PhD trainee who's getting a PhD in neuroscience and plans to go into psychiatry for medical training. And within that training process, you're sort of groomed to take positions in academia, either at a hospital or research institution. So I'm interested in hearing your thoughts on what are the role for people with this sort of training in industry, pharma, biotech, and how can we sort of expose people with this sort of training to what those opportunities might be and how to prepare themselves to be inserted into those situations?
So let me just say, this is only available to the studio audience. If you stay for the mixer, there are actually a number of people from some very strong biotech and pharmaceutical companies here, so you might ask them directly, but I know that Stacie probably has some advice for you.
I mean, I think first and foremost, there's a very, very important role for people with the kind of training you're getting to play in industry and in the development of these drugs. I think that, understanding a deep scientific understanding, but then also having that clinical training to understand how do we design effective trials? What are the best ways? What are the endpoints we're looking for? How do we design these? These are critically important that we have very highly skilled and well-trained psychiatrists in industry to help run these trials. I mean, I think that there certainly is, as Guoping said, we need more skilled psychiatrists in all aspects, whether it be in seeing patients or in developing the drugs or in studying the clinical trials. And I think there's a recognition of that. And I would encourage anyone who's interested to reach out to various companies. I think there are plenty of learning opportunities. I think there are lots of people who would be happy to be mentors, may even be some internship possibilities. So I think that, casting a wide net is always helpful, and it's one thing that you may get ignored by a lot of different feelers you put out there, but it only takes one to be the right connection. So,
good question over here.
So I want to address the question of phenotypes again. It's taken many years, and as you said, 50,000 patients to find 100 variants predicting a certain disease, but then to take it back, those were patients who were collected because they had a DSM criteria. You all only had to draw blood from them. To take it back and say they've got this variant, what's the phenotype and what's the natural history of that phenotype? What are we as a biomedical organization or society going to have to do to actually link the genetics back to the phenotype?
So I think I'm going to let Ben pick this up, but one of the things that I think as a community people are hoping to do across many diseases is to do first-order genetics in this simple way, get people identified by traditional diagnostic criteria, draw some blood, but then create cohorts that are recontactable and look at different clusters of genes and recall those people, and then it's worth deeply phenotyping. The problem, of course, is very expensive to keep cohorts going, and the ethical issues are not trivial, ranging from data privacy to creating false hopes for being in such a cohort. But what are you planning?
So I think there are a couple of strategies that we can entertain. One is this idea of passive phenotyping. So this idea that, as we interact with technology more and more regularly and that information becomes more and more available, that we could think about enrolling research subjects who would consent to providing access to those kinds of data as long as we respect the privacy and human subjects considerations, because this is like the most sensitive of sensitive data. It's, in a lot of ways, it's your life. And then the second thing that we can, I think, also consider is that as more and more healthcare providers and more healthcare systems move to electronic medical records and move to this world where your kind of medical history follows you and is actually for research, I think there's the possibility of leveraging those kinds of resources to learn about how different medical conditions relate to one another, about whether certain risk factors that we identify in one disorder like schizophrenia may also relate to other disorders. So I think those are two promising potential avenues, but it's never going to fully replace this clinical research where we enroll patients and then we want to ask very specific questions or generate very specific measures that we'll want to also eventually scale up, but I'd rather make guesses about what they are first before going through all the work of doing that up front.
But it sounds like a staggering problem.
It is. But we like the challenge.
So there's one follow-up Twitter question, but then take the microphone. Again, it goes really to this big problem, expensive amount. How do you invest effort? And somebody on Twitter asks, for academia, aside from getting an NIH grant, how do you make a go no-go decision on pursuing an avenue of basic science or preclinical research or something like this. We could ask Guoping as an academic. We could ask Stacie as somebody who's funded research.
Yeah, I mean, I think that it's different depending on what the project is. So there is no one answer to how do you make a go, no-go decision, because it's always going to be unique to the project. But anything that I'm looking at, whether it be in academia or industry, it's is what I'm studying relevant to the human disease? I think the quicker you can get some assessment of that, the better. And then I think, is it something that is druggable? Is it something that you can target? If this is something that is involved in a myriad of different processes, biological processes in the body, that if you target it to yes, you might fix the disease, but you're going to kill the patient because you've done something else. The quicker you can get a handle on these kinds of things, and the more honest assessment you can give it, frankly, the earlier you stop something that is not going to be a fruitful path, the more quickly you can get onto something that will be. And so I think it's a hard decision, but it's important.
Scientists, like all human beings, tend to find something that's work, and they kind of keep mining that same vein of coal, as it were. And there's a lot of momentum behind some areas from the pre-genetic era, right? And so we're now on the cusp of change. And what do we do actually to get the word out, or convince, or buy in, or discuss these things with the research community?
Well, I also think there's a lot of confirmation bias that goes on as well, and the way the system is set up. And this is academia and industry in many ways, in that it's to get your next NIH grant, then you need to have preliminary data. And so it's very hard to try a new idea. And I think in the same, a small
bio... You've never published on your new idea.
Exactly, you've never published on it. You don't have the data yet.
Yeah.
And you want to keep proving that it's worth continuing to study whatever it is you've been studying. And I think it's the same. If you're a small biotech and you have one pathway you're going after, one drug, you better prove that continues to be relevant to get your next round of investment, to keep your company alive. And even in big pharma, where we think that there is a kill it quickly mentality, sometimes goals are set around how many molecules did you move forward, or how many, and I think really taking a step back, and some of this may be even as reviewers on grants, as how the process goes, that we really need to be open to taking new data and incorporating it into new ideas, and understanding that we all want to move things forward in the best way, in the quickest way possible. And sometimes that means stopping what you're doing and restarting in a different way.
Terrifying. I have a question here.
Yes, actually, I would like to ask two questions. And the first one is about the basic research, because it's also my field. And you said that I like this about bootstrapping of the basic research findings. So you said it's difficult to have biomarkers because we need to do autopsies for neurological disorders. But what about finding biomarkers that are available in biopsies? Like, can we really have a biopsy that has peripheral nerve tissue, and from that either cultivate induced pluripotent cells, or find an actual biomarker in the peripheral nerve tissue, and with the new technology somehow make the biopsies have the validity of an autopsy? And that's my first question. And the second question is, like, you said that it's very difficult to measure the effect of a drug, for example, in schizophrenia, because of the nature of the disease. But excuse my ignorance, I think also Parkinson is a neurological disease, and it's very easy to measure maybe the effect of a drug there, maybe not so easy. But I think, like, if someone can focus in diseases like that are more easy than schizophrenia, can maybe applicate these more advanced techniques like gene therapy or CRISPR editing.
Yeah, so these are very good questions. I mean, the first thing obviously is both ethically, and because these are not diseases of single cells but of networks, we're not going to be doing brain biopsies for schizophrenia or depression. But I don't think that's what you really meant. We can do a skin biopsy and get a stem cell. So how about that, Guoping?
Yeah. So I think this is related
to
skin cell. This is related to both the real biopsy. For example, there are a few certain number of surgeries you can take a piece of a brain, for example, for epilepsy in some condition. But the brain is not like liver. every piece is different. So you can only get a teeny tiny piece. It does not actually, that piece may have nothing to do with autism. For example, autism, some of the kids have epilepsy, like seizures. They do, occasionally they do surgery. But there is a very huge limitation for that. However, taking the blood, skin cells or blood cells or fat cells, turning them into the neurons, and that has a potential to really look at how, what's the pathophysiology behind this. Right now, we're at a very early stage. We cannot derive robustly into all cell types. We have thousands of cell types in it, but we're moving that direction. So right now, there are a few cell types we can develop, and we can look at the major. If there's a very strong cellular disruption, we can detect them. But all these subtle things, schizophrenia, disease, maybe there are subtle changes of the neuronal function. We still cannot detect it very robustly right now. So this technology will develop, improve in the next few years. So it could end up, my prediction could end up in the future, you basically will be able to take the skin cell from patients and look at the cellular phenotype. You might be able to subgroup them. And then you can test which checkpoint is more effective, just like some of the cancer field now. So in the future, that is totally possible, and much more precision medicine compared to right now. We're giving them, antipsychotic drugs for every single one of them.
Right, and I think on your second question, did you want to say something?
No, I mean, I think that part of why we're excited is that I think we are starting to get a better handle on being able to do trials and test ideas in schizophrenia and other things. I think there's plenty of work going on in Parkinson's, Alzheimer's, that may seem more tractable, but I think they're just a little bit ahead. And I think that this is why there's so much excitement is, I think we weren't there yet in those diseases either until fairly recently, and I think there's room for lots of growth in these diseases that really affect a lot of people, and I think it's time to focus on
Parkinson's
them.
is a great example of how hard the brain is because we've known a great deal about the precise defect in the brain for a long time.
We still can't fix it.
The first drugs, L-Dopa and related dopamine compounds, date from a very long time ago, and except for surgeries like deep brain stimulation, we still haven't made the progress that we wanted to fix it. But I think like these psychiatric disorders, I think the same rules apply to Parkinson's and other neurodegenerative disorders. It really is new optimism. There's a question that just came in on Twitter asking about fMRI, functional MRI. Why hasn't that, it allows people to peer into the brain, why hasn't that given way to new forms of diagnosis?
So I think people have been trying that for a while. And I think the reason it hasn't given way to new diagnosis, at least in psychiatry, is because it hasn't worked clearly enough to be clinically relevant. And that doesn't mean that there aren't potentially components of fMRI that would teach us about certain circuitry considerations that you could then make the argument would be worth looking at as one of these intermediate phenotypes. But until we get to a point where we can show that the same genetic risk factors that influence these fMRI phenotypes also influence the outcome mental illness that we're interested in looking at, and we can kind of connect those dots, as we've been saying over and over during this panel, it's not actually going to change the way in which we diagnose these things, and it's not going to be a kind of useful, clear endophenotype for therapeutic intervention. It could work, but it's hard. It's hard to know.
What you're saying is, as we learn genetics, that becomes an anchor for any kind of brain imaging.
Yep.
And
well, we just published a paper looking at structural MRI in deep brain regions, and trying to, so there's this large-scale consortium called Enigma that works on aggregating genetic data for structural MRI, and they're moving into functional MRI. And we took those results and we lined them up against the schizophrenia results, and there was no overlap. We tried. We took 3 or 4 different cracks at it, and happy to try again with each subsequent measure that we get there. But for the, at least the deep brain region, structural MRI, that didn't seem to deliver the goods.
Okay, so question.
I'm curious if you could bring together some other communities to talk about these issues with neuroscience, neuropsychiatric diseases. What other communities do you think need to get involved? More bioinformatics, more technology? What would your dream group be?
Well, I think you kind of hit the nail on the head with the question itself, which is that the problem is too great for any one group of investigators to really take on, right? So we need not only to do our jobs as geneticists and find the genetic risk factors, but we also need to find ways of making those results accessible for biologists to perform the follow-up screenings and assays that are relevant, which can then be handed off to people that do the biochemistry to do the therapeutic development. And it's only through open lines of communication across all of these different constituencies, I think, will we actually gain any traction in the problem at all. Like my boss and mentor, Mark Daly, likes to say, academic has two meanings. One is, going to university or of a collegiate nature, and then the second is kind of pointless and irrelevant. And we don't want our kind of genetic activity to be a purely academic one. We want to take these genetic findings and make them available to the biologists so that they can start to move the ball forward in really understanding what those biological processes really are. And so I think what Steve's kind of been trying to put together at the Stanley Center is definitely a step in the right direction there. But there are other such initiatives, but it has to be interdisciplinary by its very nature.
And by the way, we also need patient groups and family groups because, frankly, without their partnership, and I really mean that, meaning long-term trusting engagement, we won't, to go back to the earlier question, we won't be able to put together the kind of cohorts we need to understand phenotypes and to see whether new interventions are really making a difference. I mean, one thing that I also discovered years ago when I was director of the National Institute of Mental Health is that when we do clinical trials, we measure symptom reduction, and of course, the funders and the regulators agree that no one disagrees that that's really central and critical to whether a therapy is working. But if you ask patients, say with depression, what they care about is, am I going to be able to go back to work or go back to school and succeed? And if you ask families of kids with neurodevelopmental disorders, they want to know, is my kid going to be able to do okay in school and have a bright future, as opposed to, decreased symptom counts. And so we have to do better, not only in terms of fixing the DSM problem, we have to find ways that are meaningful and affordable to actually give people, what they really want and need to have, to flourish in their lives. So thank you for that question. Thank you. Thank you for the audience. This has been really a lot of fun for me and a lot of great questions. I know we could go on longer. Let me thank everybody who is in the ether or has been with us online, and thanks for your questions. I'm sorry we didn't have time to get to all of them. If you know someone who missed the event today, have them visit the website. A recorded version of the webcast will be available for an additional 30 days. And then I guess like a Snapchat, we just disappear. I don't know. On behalf of Keystone Symposia, my name again is Steve Hyman, and I want to thank you again for having joined us. Thank you very much.